everyone. It's for me a great pleasure to discuss with all of you about the SARS-CoV-2 viruses and the microbiological diagnosis of COVID-19. So I would like to thank the organizers for inviting me to participate in this webinar. As you know, SARS-CoV-2 is the virus causing the COVID-19. It belongs to the family Coronaviridae, to the genus beta coronavirus, and to the subgenus sarbecovirus. It has four main structural proteins. One of them is the S protein, is the protein in the spike, is the protein in the surface of the virus that gives the form that we can see in the electron microscopy here in the top of the slide. So here probably the spike protein is the most important one. Another structural protein is the E protein, that is the envelope protein. We also have the M protein, the membrane protein, and the N protein, which belongs to the nucleocapsid protein. The genome of the virus is a positive, is an RNA with a positive polarity. And among the virus, when we talk about the pathogenicity of the virus, one important step is the interaction of the virus with the eukaryotic cell. In this sense, as you see in this scheme, the virus and specifically the S protein interact with the receptor of the eukaryotic cell. The receptor is the ACE2 protein, is an angiotensin converting enzyme 2. This is the receptor, and this, as you can see in this three-dimensional picture, there is an interaction between the receptor binding domain of the S protein. This protein in red is the S protein of the virus, and the receptor. So it's important because there are slightly differences between the S protein in SARS-CoV-2 and the S protein in the SARS-CoV, which explain probably the most affinity, the higher affinity of this protein of the SARS-CoV-2 for the receptor. Once the receptor, once the virus interact with the receptor, there is the virus, the genome goes inside the eukaryotic cell and starts off the replication process of the viruses, and finally the virus is released from the eukaryotic different virions, the different viruses are released from the eukaryotic cell. This is a replication of the virus. Another important thing is the evolution. What is the origin of the SARS-CoV-2? It seems like the origin is the bats. There is a high similarity between the rat G13 coronavirus found in bats. It's very closely related to the SARS-CoV-2. So probably the origin of the virus is the bat, and probably there is a link between bat and human. There is an intermediate, which is probably another animal, can be a pangolin or can be another type of animal that has not been described or discovered yet. When we see the scheme of the replication of the virus, as we mentioned it, we can detect several targets where we can develop some drugs to fight against the COVID-19. One potential antiviral drug can be this that blocks the interaction between the virus and the receptor, as you can see here. There is another possibility, which is the blocking the inhibition of proteases. Proteases are playing an important role in the replication of the virus. Another possibility is to find some inhibitor of the inhibition of the polymerase, inhibitor of the polymerase. There are some antivirals that can block this kind of polymerases, so it can be another target. And finally, there are two other targets which belong to the entry and exit of the viruses. So there are people working in different antiviral drugs against these targets that I just mentioned. When we talk about the diagnosis of COVID-19, we have three main approaches, three main methods that we can use to diagnose COVID-19. The first one is the use of the RT-PCR. RT stands for transcriptase reverse PCR, and there are other amplification tools. This is the first approach that we can use. And with this approach, we detect the genome or regions of the genome of the virus in the clinical samples that we are collecting, that we will see later on. Another possibility is to detect also directly from the samples the presence of the virus, but not detecting the genome. In this case, detecting some specific antigens, some specific proteins from the virus. The third approach will be what we call an indirect diagnostic or microbiological diagnostic, will be the detection of antibodies against SARS-CoV-2. And in this sense, we can talk about the detection of IgA, IgM, or IgG. There are a couple of additional methods that we can use. One of them is the whole genome sequencing of the virus. Of course, you need the specific facilities to do that. Not all the clinical microbiologists laboratories nowadays can do the whole genome sequencing, but for those that can do whole genome sequence, this is another possibility. However, there are an important difference with the previous described methods, RT-PCR, detection of viral antigens, or detection of antibodies, which is the time. So it takes time to sequence all the genome and to annotate and analyze the whole genome sequencing. There is another possibility, which is using a cell culture for the isolation of the virus. Nowadays, there are not too many labs that can use cell culture to isolate viruses. So I will focus my talk in the three first methods: RT-PCR, detection of viral antigens, and detection of antibodies. An important point in the diagnosis, in the microbiological diagnosis of COVID-19, is the collection of the sample. It's a good collection of the sample is crucial for the good result, microbiological result. There are several samples that we can collect. One of them can be the oropharyngeal swab. Another one is the nasopharyngeal swab, sputum, although not so many patients are generating sputum. And finally, the lower respiratory samples. To collecting this kind of samples, we need to use a biosafety measure so we can protect ourselves, or the people who's taking the sample need to be protected in order to be contaminated. The recovery change, or the positivity of the sample change a little bit according to the sample. For pharyngeal swabs, the recovery is 32%. For nasopharyngeal swab, it's 63%. For the sputum is 72%, and for lower respiratory samples, 92%. Once the sample is collected, mainly nasopharyngeal swab or oropharyngeal swab, these swabs are introduced in a media that can be used to transfer the swab from the collection area to the clinical microbiology laboratory. We can use two approaches to transport the sample collected. One of them is to transport the sample in a transfer media. Normally is used a media that keeps the viability of the virus. Is a transfer media for virus collection. This media, the transfer media does not inactivate the virus. The virus is still alive, so we need to take care of the sending or transport of the sample to the clinical microbiology lab. Another possibility is to collect the sample in a buffer lysis. In a buffer lysis that each lab can prepare is a mix of different compounds. It's thiocyanate of guanidine, DTT, a buffer, TRIS for instance, and Triton X100. Those compounds are inactivating the virus, so the swabs, either oropharyngeal or nasopharyngeal swab, are introduced inside of the tube with 2 mL of this inactivating buffer, and once arrive to the lab, the virus is already inactivated. Once arrived to the lab, let me show you the flow from the arriving of the sample to the lab to the generating a result with RT-PCR. We mentioned it that the samples is already inactivated. We need to extract the RNA. Remember that the genome of the virus is RNA. We extract the RNA, and the way to extract the RNA can be, we can use several ways. One of them is a specific platform for nucleic acid extraction. Nowadays, most of the clinical microbiology laboratories are different platforms to extract the nucleic acids, and they can use these platforms to extract the RNA. Of course, there are specific kits applied to each one of these platforms to extract the genome. We can use also manual extraction. This is more cumbersome because you need centrifugation, incubation, and so on. But if you are not having this kind of platforms to extract the DNA, these automatic platforms, you can use a manual extraction. There is another possibility, which is the quick and easy way to extract the RNA, which is only eating the sample, the swab from the oropharyngeal or nasopharyngeal area, eating the sample, eating the buffer at 98 degrees for 10 minutes. And that's enough. That's enough to generate a good result. Once the RNA has been extracted, we can amplify different genes. This is the genome of the RNA of the virus. They have several open reading frames, and there are several protocols to amplify different genes. The first protocol, which amplifies 3 separate regions of the N gene, as the protocol designed by the CDC in the United States. It detects 3 different regions from the N gene, and it's a protocol that has been extensively used in the United States. There is another protocol which includes the amplification of the E gene and the Rdrp genes. Both genes are the E gene is the gene involving the envelope protein, and the Rdrp gene is the gene involving the polymerase of the virus. This protocol has been designed by the Charité of Berlin and is used by a lot of labs in Europe. There is a third protocol, which is detecting the ORF1ab gene and the N gene, which is a protocol designed by the Chinese CDC. More or less, all of them are the same, has the same similar sensitivity and specificity. Of course, the sensitivity and specificity depends on the type of primers and prof selected, but in general, the sensitivity and specificity is very similar among these 3 type of protocols. We have false negative results with the PCR, and this can be associated with different factors. The first one is the poor quality of the specimen. You know that we mentioned it that it's critical to collect a good specimen, either oral pharyngeal swab or nasopharyngeal swab. So to collecting a poor quality of the specimen is can generate a false negative. Second one is the specimen was collected late or very early in the infection, during the time of infection. Another possibility for a false negative is the specimen was not handled and shipped appropriately from the site of collection to the lab. We are talking about RNA as a genome. RNA is unstable, can be degraded during the shipment, and it can generate also a false negative. And there is another possibility, which is a technical reason inherent to the PCR, and can be related either to a virus mutation, we will mention that now, or a PCR inhibition. There can be some compounds in the sample that can inhibit the PCR. Normally, the ways that we use to extract the RNA release or remove these inhibitors, so the PCR is not inhibited. And in addition, we are using an internal control to know that indeed there is not any kind of inhibitors in the PCR. Another possibility for a false negative is a virus mutation, and there are this table showing of genomes with mutations in the primary region. You can see here there are the different genes or the different regions that are amplified, and among them the N region described in the Chinese CDC protocol is the region where we can find more mutations in the primary region. So that means that the probability to get a false negative due to the fact that the primer is not hybridizing well with the region is it's higher than the other using the other genes to be detected. There are other amplification systems, or systems which are integrated. One of them is the stacked diagnostic IgN system. Is a system that this is the apparatus that is used. Is a system that you can detect all the most frequent viruses causing respiratory tract infection, and they included among these viruses, they included also the SARS-CoV-2. It has advantages and disadvantages. The main advantage that is in 1 hour you can have the result. The main disadvantage is that you have you are proceeding one by one the samples, so you cannot proceed 50 or 100 samples at the same time. So this is the main drawback of this using this system, as well as the paired express SARS-CoV-2, which involve or detects 2 genes, the N2 and the A gene. This is the apparatus. It's also an integrated apparatus. That means that you, in both apparatus, you put the sample in a cartridge, and you put the cartridge in the apparatus, and in the same apparatus, the lysis of the virus, the extraction of the RNA, the amplification, and the detection is made in the same way, in the same system. So you put the sample, you come back after 1 hour and you take the result. So it's a very good way to implement in a emergency department when you have patients that need a rapid result. This system is ideal for this kind of a scenario. There is another possibility, which is the detection of antigen. As I mentioned before, the detection of antigen, it will be ideal for a rapid detection of the presence of the virus in the clinical sample. Normally, the way that we are using the system is the lateral flow or immunochromatography. The lateral flow can use 2 approaches. The first one is the colorimetric or colloidal wall. It's in this approach, you see a band is a rapid test. In 15 minutes, you have the result. The fluorimetric system, it's you need an apparatus to detect the fluorescence of the detection of the antigen. You need a reader. In our hands, all the kits that we have tested in order to detect the presence of the antigen or the presence of specific proteins in nasopharyngeal swabs or oral pharyngeal swab, the sensitivity is really low. It's very poor sensitivity. Of course, depends on the viral load. If you have a higher viral load, 10 to 8 virus by microliter, so then probably you get a positive result. But in general, all the kits that we have tested in our lab, the sensitivity is not good enough to implement in an emergency department. Will be ideal to have a rapid test to detect the antigen in 15 minutes or in 20 minutes in the emergency department in order to decongest the amount of people attending this department. The third approach, as I mentioned, is the detection of antibodies, either EGA, EGM, IgM, or IgG. We can use also lateral flow to detect the antibodies, the presence of antibodies. In this case, the sample will be blood or serum from patients. We can also use ELISA or chemiluminescence for the detection of antibodies. So we can use 3 approaches: lateral flow that takes 15-20 minutes, and ELISA or chemiluminescence, which are most sensitive than lateral flow, but takes more time, takes at least 1 hour. The kinetics of the immune response starts around the day 5-6 after the onset of the symptoms. We will see later on the kinetics of this immune response. There is not much work done about the kinetics of the detection of IgA. the colorimetric versus fluorimetric assay is also similar to for detection of antigens. A fluorimetric assay or is a present a higher sensitivity compared to colorimetric assay when we are using the lateral flow to detect antibodies. There is a high variability among the kits in the market. There are more than 100 kits in the market that can allow you to detect the presence of antibodies in the blood or serum of patients. So my recommendation is try first, because there are sensitivity and specificity of these kits, which are already in the market. It's very diverse. Can go from 60% of sensitivity to more than 85 or 90%. Sorry, the sensitivity is low during the first days and increase to around 70% or more after the day 10. When we use the detection of antibodies for during the COVID-19, so we can have several applications of the detection of the antibodies. The first one is when we have cases that we have repeatedly negative PCR, in which we know that the symptoms have started several days before, 8, 10 days before. Then it's interesting, we can use the detection of antibodies to confirm the infection when there is a suspicion of a false negative of PCR. But again, when the patient has already passed 8 or 10 or more than 10 days after the onset of the symptoms. The second application of the detection of the antibodies is the detection of antibodies in healthcare personnel that will help in identifying those who are already immune and can return to work to care for infected patients, minimizing the risk of spreading of the virus to colleagues and other patients. So that will be interesting in this kind of health personnel to know exactly if they have already passed the infection or not. The third application will be to understand the epidemiology of COVID-19, also allowing us to know the role of the asymptomatic infections, infected people. We don't know exactly how many people are asymptomatic. In a preliminary study that has been done in Spain, the percentage of asymptomatic people in a study analyzing the seroconversion of a group of population, it's around 2.5%. 2.5% of the people has been asymptomatic. So has already antibodies, has passed the infection, but without any symptom. Another application will be to define previous exposure and identify people with highly reactive sera and can be used as a human donors for some kind of hyperimmune or this immune therapy. For research work, of course, clinical trials with this plasma of immunized patients, or also, of course, for vaccine evaluation. So if we check the evolution or the kinetics of both the viruses and the antibodies, as you can see here, before the start of the symptoms at time 0, we already have some load of virus. So in these 2, 3, 4 days before the starting of the symptoms, the patient can be contagious and can transfer the virus to the people surrounding this case. So after the time 0, the start of the symptoms, has been shown that there is a replication of the virus in the epithelial cell, and we can arrive to the level of 10 to 8 viruses by microliter of sample. So then after the day 6, 7, 8, there is a starting of the production of IgM, and there is a delay of 2 more days in order to start appearing the IgGs. After a few days, the IgM can go down but can also be maintained for a long time, and the IgG are up and also maintained for a longer time. If we analyze it, the interpretation of the presence of IgM and IgG, if we have a patient that is IgM negative, IgG negative, is probably a patient in a presymptomatic or time, or it's the first days of the symptoms, or can be also a patient that has not produced antibodies. We'll see later on that there is a few percentage of people that doesn't produce antibodies. During the initial phase, approximately after the 5-6 days, there is a production of IgMs and also IgGs. During the second phase, between the 8-day and 14 days, there is production of IgM and also IgG can start to be produced. And the third phase, more than 15 days, we have both IgM and IgG, and past infection more than 30 days, we have a past infection. People is immunized, and we have IgG, and IgMs can stay longer, can last for over time, but normally we can have an IgG positive, IgM negative, and that means that the patient has already passed the infection. So a question that we need to address, and it's, are people with antibodies against SARS-CoV-2 really immunized? There are a couple of papers that are interesting to discuss and can address this question. The first one is this paper published in Nature in 2020. They analyzed 9 cases, and they cultured the virus. They took nasopharyngeal swab, and they cultured the virus in order to see if the virus is replicating from time 0 to 14 days. And as you can see here, until day 8, the virus isolated from the nasopharyngeal swab can replicate, so it's replicating in the culture. However, there are, after the day 8, there is no replication of the virus. That means that the virus probably is not contagious, and that is related to the higher level of antibodies in these 9 cases and these 9 patients. So there is probably a link between the higher production of antibodies and no contagious of the virus, although the virus is still present in the nasopharyngeal swab. There is another paper which is in peer review. They analyzed 175 patients which are already recovered from the COVID-19. They found that 70% of the patients has a high titer, a high quantity of neutralizing antibodies. And so in principle, our antibodies are protecting the patient to acquire a second infection. 30% of the patients had a low level of antibodies, but among these 30%, 5%, that means 10 patients, didn't produce antibodies. And which is interesting is that these 10 patients without production of antibodies were young people. All of them were young people below 45 years old. And probably they are cured, so they're people that pass the infection, and probably the recovery is due to a more cellular immunity than a new antibodies immunity. So probably the recovery is associated with the production of T cell cytotoxic cells. So some messages to take at home. The first one, I already mentioned it and repeated several times. It's important to collect a very good sample to generate an optimal diagnosis. It's important for a good PCR result to get a very good sample. The PCR is still the most often used method to detect SARS-CoV-2 during the first days of the infection. Although if we have a patient that it's already has more than 8-10 days of the onset of the symptoms, we can use also as a complementary detection, the detection of antibodies. The third message is the detection of antigens would have been an ideal approach for a rapid diagnosis if the sensitivity would have been much higher. But again, the kits that we test in the lab to detect the presence of proteins, the presence of antigens in the nasopharyngeal webs were really has a very poor sensitivity. And finally, the detection of antibodies, as I mentioned, it could be a complementary assay for diagnosis if the sensitivity is over 70% and if the patient has been symptomatic for more than 5 days. And that's all. Thank you very much for your attention. It has been a pleasure for me to present this talk. Thank you.